So hello guys, welcome to my YouTube channel Med School with Shubham. I am Shubham, a third professional MBBS student, and I shall be teaching you anatomy. I have started a series on embryology on my YouTube channel in which I have completed the first, second and the third week of development. And today I shall be teaching about the placenta formation. So before starting the placenta formation, let's revise some important topics which will be required in this lecture. So the first is endometrium. So the endometrium after implantation is called as the decidua. And there are three types of decidua: decidua basalis, decidua capsularis and decidua parietalis. Trophoblast. So the trophoblast is further differentiated into the syncytotrophoblast and the cytotrophoblast. Cytotrophoblast was the one in which the cell membrane was present, and syncytotrophoblast was the one in which the cell membrane was absent. And the chorionic villi. So there are three types of chorionic villi. The primary chorionic villi, which contained only the cytotrophoblast. The secondary chorionic villi, which contain the cytotrophoblast plus somatopleuric extra embryonic mesoderm, and the tertiary chorionic villi, which contains the cytotrophoblast, somatopleuric extra embryonic mesoderm and the fetal blood vessels, and it contains the fetal blood vessels. So these are three types of chorionic villi. Guys, if you have not watched my previous lectures, I request you please go on my YouTube channel and watch all the previous lectures in which I have explained you all these topics in a quite detailed manner. So this lecture will be a very easy thing for you if you watch those videos. So I have made a diagram over here in which I have shown implantation has taken place. So after implantation, this part is called as the decidua basalis as it forms the base of the blastocyst. Then the thing which is outside to this blastocyst, this is the syncytotrophoblast, and this black color is the cytotrophoblast. I hope you remember. And this yellow color one is the extra embryonic mesoderm. Or to be more specific, afterwards the coelom will be present, and then it will be called as the somatopleuric extra embryonic mesoderm. The upper one is the epiblast and the lower one is the hypoblast. These are the amniogenic cells, and these are the this is the Users membrane. This is the amniotic cavity. This is the primary yolk sac. I hope this is very clear to you guys. Now I shall be cutting this part from here, and I will be elaborating or making the diagram of this part on the next page. See, there will be cytotrophoblast, the syncytotrophoblast, the decidua basalis, and the somatopleuric extra embryonic mesoderm. Somatopleuric extra embryonic mesoderm, cytotrophoblast, syncytotrophoblast and the decidua basalis. The same diagram I shall be making on this page now. So first, this is the cytotrophoblast, and it has formed a chorionic villi. We will show the somatopleuric extra embryonic mesoderm inside this. So I hope you can name this, which is the chorionic villi, primary, secondary or tertiary. Yes, it is the secondary chorionic villi because it has cytotrophoblast and the somatopleuric extra embryonic mesoderm. Then this is the syncytotrophoblast as I shown in that diagram. This is the syncytotrophoblast. And above to the syncytotrophoblast, we shall be having this decidua basalis. Okay. See, I have drawn the same diagram over here as this is. Just I have made a difference that I have made a secondary chorionic villi over here, an extension of the cytotrophoblast, and then extra embryonic mesoderm invaded there. So I have shown secondary chorionic villi. Rest everything is same. The secondary chorionic villi, the syncytotrophoblast, cytotrophoblast, and the decidua basalis. Okay. Now let's come here. Now this is called as a secondary chorionic villi, but it is also called as truncus cori. Truncus cori. Okay. Now what will happen in the next step? As you all know, inside the syncytotrophoblast, as I taught you in the changes in the syncytotrophoblast in the second week of development, the small lacunae are formed inside the syncytotrophoblast, and these these lacunae are then filled with embryotrophs, and then new maternal blood vessels are formed, and then they will come in contact with this lacunae, and then the fetal blood, the maternal blood will be filled inside this lacunae, and then the nutrition will be absorbed from this maternal blood. Okay, so the same thing will happen over here also. We can get the formation of the, we can see the formation of the lacunae, and here we will get the formation of the maternal blood vessels. Okay, now next thing, what happens? I hope this was very clear till the second. I hope I explained you this till the second week of development. In the second week of development, now what will happen inside this truncus cori or the secondary chorionic villi? We can see the formation of the branches in this way. So I can make it in, I can make a diagram for that. Okay, this is also invaded by the somatopleuric extraembryonic mesoderm. This is the decidua basalis, and this is the syncytiotrophoblasts. I have just made a difference that just made a change, that I have made branches inside this secondary chorionic villi. Okay, now what are these branches called as? These branches are called as the ramus cori. Ramus cori. This main branch was called as the truncus cori. Means what? This is the truncus cori, and this branch is called as the ramus cori. Now what will happen in the next step? These branches will give rise to sub branches. Okay, so it will, okay, in this way it will give rise to some sub branches. Again, this is the syncytiotrophoblasts. Now what will happen? Here you have the extraembryonic mesoderm. In this way. Okay, so let's move forward. And also we will get the formation of this lacunae. This lacunae will now increase in size, and they will form in this way. So these are all the lacunae. Okay, now what happens? And now these sub branches, I forgot to name this. These sub branches are called as the ramuli cori. Okay, so first we had the truncus cori. Then we got branches in the truncus cori, which we named as the ramus cori. And then we got the sub branches, which is called as the ramuli cori. So this is the truncus cori, this is the ramus cori, and this is the ramuli cori. Okay, now what will happen further? These are the cytotrophoblastic cells, this black one. I have not drawn the cells, but I have just drawn the line in the form of cell. So what will happen? These cells will now grow upward in this direction. In this direction. And when, see, here also it will grow from here. It will proliferate in the upward direction. And now when these cells reaches the junction of the decidua basalis and the syncytiotrophoblasts, when this migration of the cells reaches the junction of the decidua basalis and the syncytiotrophoblasts, they will now get bifurcated in the two directions. Means what? When this proliferation of cell will reach till this point, it will now get bifurcated. One layer will go in this direction and this layer will go in this direction. I hope this is clear. Now same will happen here also. When it reached till this point, when it went upward and reached till this point, it will also form two layers, one in this direction and one in this direction. Okay, in this way. And now what will happen? What will happen now? There will be a physical barrier. There will be a physical or a cellular barrier between the decidua basalis and the syncytial trophoblast, or between the decidua basalis and the syncytial trophoblast. And now this barrier is formed by these cytotrophoblastic cells. I hope you can relate. These are the cytotrophoblast. This is also cytotrophoblast, and these cells are also cytotrophoblast. Therefore, we can name this layer as the cytotrophoblastic shell. Okay, because this shell is forming a barrier between the, or it is forming a shell between the decidua basalis and the syncytial trophoblast. Therefore, it is formed by the cytotrophoblast cell. Therefore, it is called as cytotrophoblastic shell. Okay, so what happened? First we got the truncus cori or the secondary cori. This everything is secondary chorionic villi, but this is the main branch, or this is the main branch. Therefore, truncus cori. Then the sub branch. This is the sub branch. This is one branch. This is the ramus cori, and this is the sub branch of that trunk. Then branch and then sub branch. Truncus cori, ramus cori, and the ramuli cori. Okay. Then what will happen? The cytotrophoblastic cells will migrate in the upward direction or proliferate in the upward direction, and when they reach the junction between the decidua basalis and the syncytial trophoblast, when they reach the junction, they will get bifurcated in two directions, and then one will grow in this direction, one will grow in this direction. Similarly, when it reaches here, one will grow in this direction and this will grow in this direction, and then it will form a physical barrier or a shell between the decidua basalis and the syncytial trophoblast. Now what will happen in the next step? This is the decidua basalis. So this decidua basalis will now try to invade inside between these two layers. Between these two layers, the decidua basalis will now try to invade between these two layers. So these were the two layers, and the decidua basalis tried to invade inside. So now invasion will, when the invasion takes place, these two layers will now start getting separated in this way. Okay, now we will get a structure in this manner. Now because of this separation, this will move in this direction and this will also move in this direction, and a separation can be seen in this way. In this way. Okay, so now we can see the formation. Let me okay. So now what will happen here? They will get separated. Okay, and now in the center we will get the formation of this villi will be present. And here you will get the formation, you will have this lacunae inside the syncytial trophoblast. Okay, and now this because of this separation we have got this entire structure now. This one, this entire structure, this is another structure, this is another structure. So these structures are called as cotyledon. This is called as cotyledons, and there are approximately 15 to 20 cotyledons in the placenta. So this has occurred, the placenta formation has taken place here, and I hope you can see this cord like thing in this lower part. This is the umbilical cord. So let's make that one more time over here. So this is the placenta now. This entire thing is a placenta, and this is the umbilical cord. So what things we can find here more? So we will find we will get the formation of these blood vessels. Fetal blood vessels inside this. Okay, and also okay. Now let's name the structures. So this is the umbilical cord. This entire thing is the placenta. Okay, and this thing is the cotyledon. This is the cotyledon, and here you have the decidua basalis, which has here invaded inside. And one more important thing, that this decidua basalis is the part of the maternal side. Therefore, this is the maternal side of the placenta, and this is the fetal side of the placenta, or the maternal surface of the placenta, because it is facing the decidua basalis, and this is the fetal surface of the placenta, as it is facing the extraembryonic mesoderm. Okay, see, here we had the extraembryonic mesoderm. This was the extraembryonic mesoderm, and this is the decidua basalis. So this part is towards the maternal side, therefore it is called as the maternal surface, and this part is towards the fetus, therefore it is the fetal surface. And now the cotyledons are formed in towards the maternal surface, and the cotyledons will be formed towards the maternal surface, and there are 15 to 20 cotyledons on the maternal surface. Now what are the contents of the umbilical cord? So the umbilical cord contains these two umbilical arteries. I am right. These are the blue color ones are not veins. Those are umbilical arteries, because in fetal circulation we draw the arteries in blue color. Umbilical arteries, and these red color ones. This red color one is the umbilical vein, the umbilical artery and the umbilical vein. Okay, also if you are okay, let it be. So this is the umbilical artery, and this is the umbilical vein, and this entire thing is the umbilical cord. So why we have drawn umbilical arteries in blue color, and why we have drawn the umbilical vein in red color? So the reason for that I will be talking about in the fetal circulation topic. In that topic I will explain you why I have drawn the umbilical artery in blue color and vein in red color. So please stay, watch that video also. I will be uploading in some days that video. So I hope this was very clear to you. This is very clear to you about the placenta formation. Let's revise everything once again. The endometrium is called as decidua after implantation. Three types of decidua: decidua basalis, decidua capsularis, decidua parietalis. Trophoblast are of two types: the cytotrophoblast and the syncytotrophoblast. Chorionic villi are of three types: the primary chorionic villi, secondary chorionic villi, and the tertiary chorionic villi. Okay, the first we will get the formation of the truncus cori, then we will get the formation of the ramus cori, and then the framuli cori. Then what will happen? Lacunae will get formation. Lacunae formation will take place over here. Over here, the blood vessels will form of the maternal blood vessels, and inside this the fetal blood vessels will be formed. Okay, now the cytotrophoblast will migrate in the upward direction, proliferate in the upward direction, and when they reach the junction between the decidua basalis and the syncytotrophoblast, they will get bifurcated in two directions. And now this decidua basalis will now start invading in that, leading to the separation of these two layers. These two layers will now get separated, giving to the formation. It will lead to the formation of cotyledons. Okay, so this all makes the placenta, and this is the umbilical cord. what things umbilical cord contains? It contains the umbilical arteries, umbilical vein. Another two things are included inside the umbilical cord. I will be teaching you about that in the GIT chapter, in the embryology of the gastrointestinal tract. But in the notes, I shall be writing those names, but the explanation for that I will be giving in the GIT embryology. Because if I tell you now, you won't understand that. Therefore, stay tuned till that point. I will explain you everything. So thank you guys for being with me till this point. I end the video over here. In the next part, I will be talking about the clinical aspect of the placenta, the functions of placenta, and the types of the placenta, that is placenta previa. So thank you so much guys. Please like my video, share my video, and also subscribe to my YouTube channel. Thank you so much guys.